Let us discuss in detail the transcriptionization. The transcription general transcription factors collectively perform the function which is performed by the sigma in the bacterial transcription. That is, recognition of the promoter is done with the help of sigma in the prokaryotic system. Similar manner, various transcription factors are associated in binding up of RNA polymerase to the promoter and melts the DNA, and it opens the complex of DNA, double helical DNA, to form the transcription bubble. They further help the polymerase to form the further helps the polymerase to escape from the promoter and switches over to the elongation phase. These complete set of transcription factors and polymerase bound together at the promoter site indicates the process of initiation. This is called as pre-initiation complex. The RNA polymerase 2 promoter contains data elements which is found nearer 30 minus base pair upstream from the transcription start site, where the pre-initiation complex formation begins. Initially, the data element is recognized at the minus 30 region by the transcription factor called TF2D. The TF2D binds to the Tata DNA sequence is called TPB, Tata binding protein. The other subunit in this complex are called TAF, that is Tata box associated factors. The Tata box associated factors recognizes other promoter elements which are found in the eukaryotic system, such as INR, DPE and DCE. Although the strongest binding is between TPB and Tata only. Thus, TF2D is a critical factor in promoter recognition and pre-initiation complex formation. The Tata box binding protein DNA complex provides a platform to recruit other general factors and RNA polymerase itself to the promoter region. The protein which are assembling at the promoter region is in the order: once the TF2D is bound, followed by TF2A, 2B, F together with the polymerase, then joins. Then later TF2E and H comes to this position of pre-initiation complex and followed by the promoter is melted. Unlike a prokaryotic system, the hydrolysis of ATP is necessary for melting up of hydrogen bonds between the template DNA and the non-template DNA. In the eukaryotic system, the hydrolysis of ATP is done and it is mediated by TF2H to form the open transcription bubble region. Here is the diagram, shows the pre-initiation complex formation and how the carboxy terminal end of the RNA polymerase containing heptapeptide is undergoing phosphorylation during the process of pre-initiation. That is shown as a diagram. That is, the carboxy terminal end of the RNA polymerase 2 contains the heptapeptide sequence. Hepta means 7 peptide sequence, that is tyrosine, serine, proline, threonine, serine, proline, serine. Once it is polymerized, the RNA polymerase is responsible for the start of the initiation. That is, it locks at the +1 position of the start point of transcription, then it moves in the forward direction. During the process of initiation, that is synthesis of RNA, short segment of RNA is synthesized, but at the same time what happens? The polymerase moves in the backward direction until it perfectly locks at the +1 of the start point. During the reversed movement of this RNA polymerase, it degrades the short segment of RNA by the process called as abortive initiation. Abortive means degradation of RNA is called abortive initiation that is taking place. Why this is means? To lock perfectly at the +1 position of our start point of transcription. Once it is locked perfectly, the largest subunit of the the largest subunit of the polymerase II is responsible for carrying out the polymerization activity. Normally, the largest subunit of polymerase II contains carboxy terminal domain CTD, which is also known as tail region. This CTD domain consisting of 7 amino acid sequence called as heptapeptide sequence of tyrosine, serine, proline, threonine, serine, proline, serine. There are 27 such repeats are found in the case of yeast RNA polymerase II in the carboxy terminal domain, whereas in the worms it is 32 such repeats are found, and in fly Drosophila 45 such heptapeptide repeats are found, whereas in human it is 52 such repeats are found in the case of the carboxy terminal domain of the RNA polymerase II. The number of repeats seems to be correlating with the complete complexity of the genome. So, by comparing, when compared with that of yeast, our human is having 52. This is a huge number, double the size which is present. It indicates the complexity of our system. Further, each repeat containing sites for phosphorylation, that is added with phosphate group with the help of kinases, that is further assisted by TF2H. Once the RNA polymerase II is recruited, it is unphosphorylated in nature, then further these kinases are responsible for phosphorylation process to form the pre-initiation complex for the start of initiation. Then when it perfectly locks, after the abortive initiation, it perfectly locks at the +1 position of the start point of transcription, and it moves in the forward direction 5' to 3' direction for the production of the RNA. It keeps on going by the addition of nucleotide. The nucleotides are added according to the template DNA, which is complementary to that of the template DNA. For example, if the template DNA contain adenosine, means the RNA will contain uracil in that place. Here the table shows the list of transcription factors that are assisting RNA polymerase II in the initiation process. Here the details, the order of the binding up of transcription factors that are occurring in the initiation complex. Further, the transcription initiation requires additional proteins that mediates the complex. What are all the things means? The mediator proteins, that is DNA bound activators, chromatin modifying and remodeling factors are also part of the transcription machinery. What is it? Chromatin modifying and remodeling factors means, since the eukaryotic system containing DNA is associated with histone subunits that has to be eliminated during the movement of RNA polymerase in the forward direction, that are mediated by chromatin modifying and remodeling factors which are needed for it. Further, the RNA polymerase II containing carboxy terminal domain, that is a tail, is bound by the activators that pulls the RNA polymerase in the forward direction for the synthesis of RNA during initiation process. This assembly of pre-initiation complex in the presence of mediator, nucleosome modifiers, remodelers and transcription activator. Once the length of the RNA is achieved in the initiation process, about 12 to 13 nucleotides, it switches over from the initiation to the elongation phase in the case of a transcription process. How it is possible means, that is once the RNA polymerase escapes the promoter sequences and the initiation of transcription, it shifts to the elongation phase. The elongation process in which the initiation factors are fallen off and it is replaced by a set of factors called elongation factors such as TF2S and SPTS are responsible for the elongation. That is, stimulation factors are responsible for coming and replacing the role of initiation factors. These elongation factors further increases the length of the RNA. At the same time, the phosphorylation form of the carboxy terminal domain is present that binds with the elongation factors for further processing that switches to the elongation step. And additional proteins are also mediating elongation for the RNA polymerase, that includes kinases and protein TEFB. It is recruited to the polymerase by the transcriptional activator. Once the bound RNA polymerase 2 is phosphorylated, the serine residue at the position second 2 of CTD repeats. The phosphorylation event correlates with the elongation process. In addition, the protein TEFB phosphorylates and activates another protein called as SPTS. This SPTS an elongation factor. Finally, it recruits another protein called as TAT SF1, that is also recruited by protein TEFB, that switches the RNA polymerase from the initiation to the elongation phase. The direction of movement of the RNA that happens in the forward direction. Here the diagram shows the recruitment of various protein at the CTD tail of the polymerase for the elongation step. That is, the RNA polymerase recruited at the tail of the RNA processing recruited by the carboxyl terminal domain of the RNA polymerase. And here is the list of factors shown in the diagram, how it is phosphorylated at the serine position at 5 after the escaping from the promoter sequence. It is associated with the capping factors at the serine 2 position, and the addition of the elongation factors as well as splicing factors further pulls the RNA to the elongation step. The elongation factor HSPTS is also recruited at the tail of the phosphorylated serine. During this elongation phase, the hurdle for the movement of RNA polymerase is associated with the presence of histone in the DNA. The nucleosome containing histones has to be removed. For that, the fact that is associate that identifies the histone. A fact is called as facilitates chromatin transcription. Factors facilitates chromatin transcription is called as fact, that is responsible for the removal of chromatin which is present in the forward direction. This fact is a heterodimer. Heterodimer means two different domains are present which contains the conserved proteins SPT16 and SSRP1. Normally, the DNA of the eukaryotic system is wrapped with the nucleosome, that is histone protein. It's an octamer made up of H2A, H2B, H3 and H4. The histones are arranged in two modules, that is H2A and H2B dimers, H3 and H4 as tetramers. Ahead of this transcription, transcribing RNA polymerase, the FACT is removing one H2A H2B dimer. This allows the RNA polymerase to pass the nucleosome. The FACT also has a histone chaperone activity which allows to restore the H2A H2B dimer to the histone hexamer immediately behind the processing polymerase. That is, in the forward direction, it removes the dimer, and the removed dimers are further recruited at the back of the DNA that is at the back of the RNA polymerase during the elongation process. Here is the diagram, the model of the FACT-aided elongation phase through the nucleosome. We can see that H2A H2B dimer is removed in the step 1, and the RNA polymerase is moving in the 5' to 3' direction, and the FACT, what it is doing further, it recruits the dimer of H2A H2B in the second step. The elongation process of the eukaryotic system further is recruited with the RNA processing. There are various types of RNA processing is taking place. That is the case of the mRNA, that is involving capping, splicing and polyadenylation at the 3' end of the RNA. The most complicated of this is splicing, whereby the non-coding regions are removed and the coding regions are spliced together to form the mature RNA. Further, it adds cap, that is 7-methyl-guanosine cap, and many polyadenosine residues are added to the primary transcript to form the mRNA, which is mature mRNA. The elongation factor SPTS is responsible for recruiting the 5' capping enzyme as to the CDT tail of the RNA polymerase, and further HSPTS stimulates the capping of enzyme activity. In another case, the elongation factor TAT-SF1 recruits the components of splicing machinery that is responsible for the splicing up of the introns, and thereby the elongation of the RNA continued with the splicing mechanism as well as addition of cap and tail that are interconnected. Once the cap is added, cap and tail is added, the RNA becomes mature. The detailed process of splicing followed by cap addition is seen in the later. That is, normally to the 5' end of the mRNA, 7-methyl-guanosine cap is added. Then further, the next nucleotides are methylated with the help of the S-adenosyl methionine, that is the donor of the methyl group. Then later, through the 3' end of the RNA, A polymerase that adds the many poly A nucleotides that protects the mRNA. It increases the half-life of the mRNA in the eukaryotic system. This is done with the help of the machinery of capping machinery that is recruited by TAT-SF1. Here is the structure of formation of the cap in the case of the eukaryotic system. That is, the gamma phosphate of the 5' end of RNA is removed by the enzyme RNase triphosphatase. Then later, the guanyl transferase is responsible for the transfer of the guanosine methyl group to the 5' end. It forms a 5' phosphodiester linkage. Then Once the guanosine is added, the methyl transferase comes, that is responsible for the addition of methyl group from the donor S-adenosyl methionine. Then after the donation of methyl group, the S-adenosyl methionine becomes S-homocysteine. The final processing of RNA is done by the polyadenylation at the 3' end of the mRNA, that is mediated by the poly-A polymerase. Once the polyadenylation is over, the polymerase reaches the end of the chain. It encounters the specific sequences that are necessary for the termination of transcription. There are a series of events that are occurring at the transcription termination process. The transcription termination is linked to the RNA destruction. It's a highly processive. RNase is responsible for this mechanism. That is, in the case of the yeast rat1 and human XNR2 loaded to the end of the RNA, another protein RTD103 that binds to the CTD of the RNA polymerase. The rat1 enzyme is very processive and quickly regrades RNA in the 5' to 3' direction until it catches. This is similar to the Rho protein termination that catches to the RNA polymerase. That is, it moves along with the RNA in the forward direction and it reaches the RNA polymerase and pulls out the RNA for the process of termination of transcription by wrapping around it. There are two types of termination occurs in the case of the eukaryotic system, that is torpedo model of termination. Another one is allosteric model of termination. According to the termination model, it undergoes a conformational change in which the elongation process of the polymerase comes to an halt by spontaneous termination, in which torpedo as well as allosteric model mechanism that is taking place. The two proposed model of termination of transcription is shown in the above diagram. In the torpedo model, RNA transcribed downstream from the poly A site is attacked by the 5' to 3' RNase. This is called as torpedo enzyme, which is loaded to the transcript from the polymerase itself. When this exonuclease catches up the polymerase, it triggers the dissociation of the DNA template and termination of the transcription that is taking place. Whereas in the allosteric model, the polymerase is highly processive within the gene. Once the poly A signal is passed, it becomes less processive, it slows down. This alteration is due to modification or conformational change. Even in this allosteric model, the second RNA would be degraded by the RNase only. That cannot be the cause of the termination. The RNA degradation is involving the passing up of the poly A signal. That is another factor that causes termination, called as allosteric model of termination in eukaryotic system. So far we discussed the details of the initiation, elongation and termination process of transcription in the eukaryotic system. We will see the splicing mechanism of introns found in the RNA further in the next video.